So I know that there are forces afoot that are going to move us out of 146 permanently for the rest of the semester. I don't know that we'll end up here, and that's not great for me, particularly for those who facing with your back to this darn thing. But I'm just letting you know, waiting to hear what they're going to work out. Maybe we'll stay there, I don't know. But I know that they're trying to find a new room for us because of another group that wants to meet there for business purposes. So when we talk about cell division, we have to talk about chromosomes, genetic material. Chromosomes are made of chromatin. So basically, a chromosome means a colorful body. There's a picture over here makes us think, well, those are pretty colorful. They're not looking as bodies per se, but body sort of mean generically the central portion of something, what's made of. But those are chromosomes, and they've been uniquely stained in a way so that the person who was looking at these could match them up and find what are called homologous pairs. Whereas humans have 23 pairs of chromosomes for a total of 46 chromosomes. Two of those chromosomes are called autosomes. There's not really a good explanation for this, how much I tried. Basically, it's referring to the chromosomes that have to do with anything that doesn't have to do with gender. Which is, I don't know, sort of a disservice to it all, because the chromosomes that have to do with gender, at least one of them, also have a lot of important information for the body. Things like color vision, for example. But that's the way it is. The 23rd pair is considered sex chromosome. This is what they call a karyotype. If you take a cell or cells from an individual and then you put them under the conditions necessary for them to go into mitosis, where we're about to talk about, the genetic material will duplicate and condense down, and then you can put the cells under what's called a hypotonic condition, which we should be familiar with now. And then you drop them onto a slide, and the cells will burst, and the nucleus breaks open, and the chromosomes come out. They get splatted on the slide. And then you go and they capture the different chromosomes with a picture, and then you can use a computer to cut and paste. You can move the chromosomes around so that all the pairs match up. And so this has all the 22 chromosomes plus the pair of gender-defining chromosomes. And the pair of gender-defining chromosomes in this case are X and a Y, which means this person was male. The true sex chromosome here is actually the Y chromosome, because this really does have to do with almost nothing else but gender. The default condition for humans is actually female. If you don't do anything else to change the programming, you get female. The Y chromosome brings in a little region called the SRY region, the sex determining region. Or called, sorry, your male region. Anyway, it basically changes the programming and turns the individual male. The other big chromosome is the X chromosome. So if this was female, we'd have two fairly large chromosomes here instead of this short little chromosome, the Y chromosome. It's short because there's nothing else on there but gender-defining characteristics. You see that the largest chromosomes are like 1 and 2, they're quite big. And then you get some really quite small chromosomes, you get here the chromosome 21 and 22. It makes you think about the evolution of chromosomes and why some are so big and why some are so small. Some geneticists have suggest that a lot of evolution for multicellular organisms with chromosomes has occurred because of chromosome fragmentation and recombination. For instance, they often talk about us having our DNA being almost 98% identical to chimpanzees, but the chromosomes, the number of chromosomes are quite different, and that may have something to do with why chimpanzees are more different than us in terms of numbers. So a karyotype is just basically looking at the chromosomes of an individual. Karyo is almost always used whenever you're referring to stuff that you find inside the nucleus. I thought I'd bring a sort of real-world subject matter to make karyotypes more interesting. There's a procedure called amniocentesis. Have you heard of it before? If a woman is 35 years or older, sometimes her doctor will recommend to her if she's pregnant to have this procedure done. And what they do is they take a very long needle and they go into the uterus and they penetrate the amniotic sac. And that's because the amniotic sac, even though it's not technically part of the baby, well, technically it arose from the same cells that the baby arose from. They start with a single cell when sperm and egg come together, and then they start dividing, and some of those cells, by mitosis by the way, and some of those cells will ultimately develop into the baby, and other cells will become part of the amniotic sac that surrounds it, the placenta, structures that ultimately don't are part of you, and yet, genetically, they're identical to your cells. So they'll collect some of these cells in the amniotic fluid. The baby has, at a certain period in time, has skin and will start to shed these cells that it's no longer using, and they collect those cells so that it doesn't harm the baby. And then they will pull out those cells and put them under the conditions of mitosis, dividing, so they get these nice duplicated chromosomes, and then they will splat them out on a slide, and they will look at the chromosomes and they will say, are there any genetic abnormalities for this baby? Because for a woman who's 35 years or older, her chances of producing an oocyte that hasn't completed meiosis properly and has ended up with an extra chromosome 21 is higher. There's a higher risk of that happening. And the baby would then develop with Down syndrome if that's the case. So they want to know that information. There's also other possible chromosomal defects that can be detected with this too. These are duplicated chromosomes. When you see chromosomes, they're always under the duplicated condition. If we were able to take an unduplicated chromosome and condense it down, it would look something like this. Only one stick. But these are more than one. They are two, side by side. So when the chromosome duplicates, it makes an exact copy of itself. I'm not able to draw exactly. They're exact copies of each other. These are twins. But when you have this condition, you have to have, well, yeah, they have basically a name for these. They call each arm of the chromosome a chromatid. And what's more, they call them sister chromatids. And altogether, though, the whole thing is referred to as a chromosome. I honestly think that whoever came up with this was really trying to confuse people. I have to share my confusion at this point, but nevertheless, it's what they call individual arms of duplicated chromosome. They call them chromatids. We'll use that terminology appropriately as we go through this process of mitosis. And then in the center is the centromere. This is all been labeled for us here, but I put it bigger on the board for you. And we'll see that the centromere is important in the process of mitosis. Another reason to talk about it. Any questions? cells actually spend most of their time in the condition of this interphase. In fact, for some cells, they never leave it. They just stay there forever. Interphase itself can actually divide it into separate phases if the cell is getting ready to go into this whole mitosis rigmarole, this cell division that we're about to talk about. G1 is when cells will actually duplicate their organelles. They'll double their organelles up. That means they get more mitochondria, they get more ribosomes, they get more endoplasmic reticulum, they get more centrioles. Those are important. It's called the G1 or GAP1 phase. G stands for growth. It actually stands for GAP. There are some cells in the human body that never ever go beyond this phase. They stay there forever. Mature muscle cells, mature cells of the nervous system, like the brain. You have them, you produce them, and that's it. They don't produce anymore. They don't divide. They don't go into mitosis. If that's the case, we call the condition that they're stuck in G0. So, as an example, some mature muscle and nerve cells do not divide and stay in G0. A bit of a run-on. English professors will have to forgive me. So G0, they're never going to divide. They're stuck there. However, every other cell is going to go into mitosis, then enters into another phase. S for synthesis. And the synthesis is mostly focused upon DNA. So during the S phase, we synthesize DNA. And the centrioles that they haven't been produced yet by the way. So at that point, chromosomes are duplicated, so we end up with producing chromatids. And that's what they're showing in this image above here. There's an individual unduplicated, and we go through duplication or replication, and we end up with two arms and two sister chromatids, duplicated chromosomes. So G2 is more growth, final preparation for what's really an energy-intensive and stressful and prone to mistakes mitosis. I should say, prone to. There's a lot of mechanisms in place to make it so that mistakes aren't made during mitosis, but nevertheless, it is when cells are probably their most vulnerable in terms of the possibility of producing a mutation or making a mistake because of this, all the rigmarole in terms of moving chromosomes. In fact, even during the S phase when DNA is replicated, there's the opportunity for mistakes in DNA replication, which could cause a problem with the cell in the future. So mitosis and meiosis are all about nuclear division. They're about dividing the contents of the nucleus. I don't want you to think we're talking about atomic bombs here. Dividing the contents of the nucleus. Which is really mostly about the DNA, about the chromosomes. see that cytokinesis, which literally means cell division, is just a little sliver here, just the last part of it. So we take cytokinesis, the word cyto means cell, and kinesis means to divide. That's when true cell division occurs. All the rest of this, let's get ready for that one moment. All mitosis is not how we separate genetic material. So cytokinesis, the way of looking at it is cytoplasmic division. So do cells just divide whenever they feel like it? I mean, if they did, wouldn't we end up with too many cells? We just basically keep dividing and growing and growing and growing, and yet we grow to a certain size, and for the most part, not counting things like adding more adipose into adipose cells, we don't grow anymore. We get a certain number of cells, and we just kind of max out there. And any growth, we go to the gym, or work our muscles, or you just eat a lot of food and gain a lot of weight. Any of that kind of growth is almost always about increasing the size of the cells that you have, not increasing the number of cells that you have. So the number of cells we have are tightly regulated and controlled. When you start growing numbers of cells beyond that top number, whatever it might be, when that starts happening in some place uncontrollably, that's what we call a tumor. Have you ever heard of a benign tumor? Then you also heard that word cancer. It's not the same thing. A benign tumor refers to some cells that decide to grow and grow and grow and create this mass, which, by the way, can be life-threatening. It's in the wrong place, like your brain. Versus a cancer, which is a bunch of cells growing uncontrollably, but also have the additional ability of migrating to different parts of your body and starting new tumors and growing uncontrollably there. They call that metastasis. That's when it becomes cancer. So there are tight controls in terms of how cells grow in order to avoid that possibility. If there are too many cells, we start producing too many cells, there's actually even a mechanism within our body that tells cells to die. We call it programmed cell death or apoptosis. Cells, particularly immune system cells, can trigger other cells to death. And that will reduce inflammation too. But when cells die that are not programmed, they just burst. That causes inflammation. The immune system gets all worked up. So what's going on here? There's damage here. We're not in charge of it. So there are a number of checkpoints during cell division, during the cell cycle, in which the cell can check to see if it's ready to continue on with its growth, its division. So during gap 1, there's a check for growth factors to see if the DNA is intact. If it's not, then there might be steps in order to repair that DNA. And if that's impossible, then maybe the cell will be told to die. We'll have a mechanism, an internal mechanism, we don't need the immune system, that will basically trigger cell death because it's broken. And then you don't want to keep it around because it can lead to other broken cells if it were to divide. Or maybe we just don't let it divide. We just stick it in G0, keep it around because it's doing a job, but we don't kill it. But we don't let it divide either. So there are all those possibilities if the DNA is not intact, the growth factors are. During G2, at that point, synthesis should have occurred and we should have duplicated DNA. If we don't, then we should not go into cell division. So the cell is checking that. Did the DNA get duplicated correctly? If it did not, then we are not going to go into cell division. Is the DNA damaged? If the DNA is damaged, then we may tell the cell that it needs to die. We can't repair it. If we can repair the DNA, then we can move forward with cell division. into cell division because what makes the cell tumorous or cancerous has to do with when it is got damaged DNA that allows it to divide uncontrollably. So in order to avoid that possibility, cells that have damaged DNA are often either repaired if they can, or just told to die for the good of the rest of the body, because it's just problematic. And there's ample evidence for why this is important. In conditions or illnesses where we have chronic illness, for example, ulcers in the stomach, we brought that up earlier in the semester, or chronic liver damage and cirrhosis, the chances of actually developing a cancer are even greater because with that damage, the way the organ will try to repair itself is by making more cells. So it engages in more mitosis, which means there's a greater chance for producing DNA that's damaged. Because anytime you go into mitosis, there's always that chance. And the more mitosis you have to do, there's the chance every time that you're going to produce damaged DNA and a damaged cell. So a tissue that suffers this chronic damage over time is more likely to produce a cancerous cell during that time period. That's one of the reasons why trying to avoid those conditions. So take care of yourself. Less likely to get cancer, but it's always a possibility. Even during mitosis, there are checkpoints. During metaphase, particularly between metaphase and anaphase, the cell will make sure that its spindle fibers have attached to the centromeres. So that's kind of the weird thing about this lecture is I have to talk about things that we haven't really looked at yet in cell division. So when these are made, the idea is that we get one of these for one cell and the other one for the other cell. In order to move those, we have to attach to them spindle fibers made of our good friend tubulin. I want to make note of this because it will be important when we are doing our lab today. Bringing those spindle fibers into play. So we have another spindle fiber over here. And the idea ultimately would be to pull these apart. We want this chromatin to go that way, that chromatin to go that way. And then they are going to go to two separate locations that will become two separate cells. So if this attachment isn't done right, then we end up with too much genetic information in one of those cells. If this attachment is broken, then both of these go to the other cell, and this cell is now lacking a chromosome, which means it may malfunction, may become a cancerous cell. So to make sure that the spindle apparatus is good attachment before moving forward. So a number of control mechanisms, a number of checkpoints to make sure the cells are fully ready to go into cell division. Questions or thoughts? There's even external control of cell division. As I was talking about, we get a certain number of cells in our body, and pretty much that's it. Those are the cells that we have to work with. We can replace cells, but the number doesn't vary a whole lot. And part of that control is exerted by other cells. You see, if in this picture up in the upper left-hand corner, we have a signaling cell releasing a chemical signal, and this receiving cell has a receptor for it. And then there's a transduction of that signal across the cell, and ultimately that activates some proteins inside the cell that turn around and activate some DNA. You get gene activation, you get production of messenger RNA, and it gets turned into protein. So later we'll be talking about transcription and translation and how that works to make new proteins. In terms of making a new cell, this is what you have to do. So some of those signals are hormones, estrogen, progesterone, testosterone. All of these are called anabolic steroids for a reason. They stimulate growth. They stimulate the production of new cells. So progesterone can stimulate the uterine lining to increase its number of cells that thicken in preparation for the implantation of a fertilized egg and ultimately a baby. And then non-hormonally, epidermal growth factor in the skin can be released in order to stimulate skin growth. So if you change jobs from being a secretary to going out and working construction, and you're having to lift a lot of heavy stuff all the time, and you're using your hands more, and there's a lot of abrasion. Can I ask why we have phones and hands on the body? Because I really don't want that.
Because I can't see them.
Because you can't see it.
Yeah, these are pictures. I'm sorry.
So these pictures are available online. You know that? Yes. On Canvas, the whole thing. This is also available in the textbook. The outline too. So when you stimulate those areas of the skin, when you get that abrasion going on, then the skin actually will thicken in those areas in order to give you some additional protection. And epidermal growth factor is a signal that's released in order to stimulate those skin cells to grow. Following calluses. So signal transduction is just referring to the fact that after the signal is received, there has to be a way for that signal to be then sent to the protein, and that's a signal transduction. So there are a number of genes that are involved in controlling cell replication, and that can both stimulate or inhibit the cell cycle. Proto-oncogenes are often classified as stimulators of the cell cycle. I want you to look at this term for a minute. It's hyphenated. Proto-oncogenes. So proto is like a prototype. It's like it's not there yet. It's kind of a weird name because onco refers to cancer. And so these are cancer genes. And so we have these genes in our cells that are called proto-oncogenes. They're not cancer genes in our cells, do we? I mean, that doesn't make a lot of sense. They decided to call them this because the idea was that these genes, although they aren't causing cancer normally, they have the ability to do it. They call them proto-oncogenes because they're not truly a cancer gene yet, but if something changes them, they become a cancer gene. And there's a few of them. I'm going to get a couple of examples to talk about. And they often go under the classification of tumor-suppressing genes. So normally these genes, when they're normally working, are referred to tumor-suppressing genes. They actually inhibit the cell cycle. And examples of this include P53 and BRCA1. So P53 is an interesting gene, something that's been studied for a long time. And we know that it's a gene that is one of the most common genes to be mutated whenever we're looking at cancer cells. They look at a cell that's become cancerous, they look at what's changed about that cell. The first gene they're going to look at is P53. And that's because P53 is like kind of a master switch in terms of controlling cell mitosis, or in terms of controlling cell division. It controls the whole cell cycle, decides when the cell divides, it looks when it basically is responsible for activating the genes that are necessary to repair any mutations in the DNA. It can also activate pathways that will lead to apoptosis. There's actually a close association between P53 and BRCA1, in fact. P53 is sort of a master class gene, it's the one that's in control. BRCA1 is a gene that was discovered when they were looking at breast cancer. This was basically BR breast, CA cancer the breast cancer, the first breast cancer gene that's described as BRCA2 also. BRCA1 they discovered, and they found that it was a gene that was critical in repairing mutations, repairing damage to the DNA. And so either one of these genes, if there are mutations within these genes, so normally P53 controls the cell cycle. So if you have a mutation in it, it's not controlling the cell cycle anymore, and the cells can start dividing uncontrollably. Now you have a tumor. If cells are dividing uncontrollably, the chances are that they're going to produce a mutation in their DNA increases because they're growing so much. And every time you produce a new cell, there's a chance for a mutation. So there's a chance, an increased chance of producing a cell that has the necessary changes to metastasize, to go from one place in the body to another place in the body. And at that point, you now have a cancer. So it's like a stepwise progression. That's one of the reasons why they want to get benign tumors out if they can. Because tumors with this uncontrolled growth are more likely to produce an actual malignant tumor later on. It can become malignant if it's not controlled, just by producing that last mutation before it metastasizes. So damage to P53, you lose control of the cell cycle, you get increased mitosis, and you get an increased chance of producing a cell that is mutated enough to actually form cancer. Damage to BRCA1 means that you no longer can repair damage to DNA. So any damage to DNA is going to stick around. And so usually when there's a mutation in BRCA1, the chances of breast cancer increases significantly. And the same thing for P53. With damage to P53, the chances are you're going to have a tumor and potentially cancer in your lifetime. Increases multiple times, multiple levels. So again, there are changes in those genes, and all these others that I haven't named, like the proto-oncogenes, you can get cell cycle, you get mutations there, and they start stimulating cell cycle when they're not supposed to, then you get a tumor. They'll start control. If you get damage to genes that normally would suppress tumor formation, then you're more likely to get a tumor. So it's a two-switch system. You have a system where you stimulate cell growth when you need to, and you have a system when you can inhibit cell growth or cell cycle when you need to. And if you damage either one, you can get more growth than you should have. Right? Too much stimulation, too much growth. Not enough inhibition, not enough suppression, too much growth. It's a delicate balance that you have to have both sides to get it just right. So as we go into this, these are just some of the factors that we want to consider about mitosis. So mitosis overall, why is it important? It's important for tissue repair. Anytime we have damaged tissue, we need to make new cells to repair that tissue. We use mitosis. So, cut on the skin, we use mitosis. Now, there are other mechanisms that are involved in repair. And that's why we get scars, because it's not a perfect replacement of all our cells. We get deposition of fibers. That's not cellular at all, but it helps hold the tissue together that was damaged. And then there's some cells that are put in there too. But mitosis is definitely still part of that process. It's important for growth. How do we go from being a baby to a full-grown adult? How do we go from a single cell to being a full-grown adult? You go from one cell to trillions of cells. That requires a lot of growth. Maintenance. So there is a breakdown, a slow breakdown of tissues inevitably. You have to replace those lost members. You have to replace those lost cells when there's a breakdown. So constantly maintaining tissues by making new cells. And then again, repair. Oh, I got redundant there, but so it's important for repair. The process of mitosis, this is important because it's going to allow us to differentiate it from meiosis, is basically we start with a single cell, and we end up with two identical cells at the end of the process. So you start off with up here because this one has unduplicated chromosomes. And by the time we're done, we have two cells that are identical to it with unduplicated chromosomes. So we're going to bring up some terminology. It's biology, so of course we have to bring up some new terminology. One of the terms we're going to use is diploid. And the other number or denomination you're seeing there is 2n. The idea is that diploid, di means 2, means that basically all of our cells have 2 chromosomes, have pairs of chromosomes. They have 2 chromosomes for any given feature. So we have 2 chromosome 1s, 2 chromosome 2s, 2 chromosome 3s, and those chromosomes, and we're going to use this term also, are called homologous chromosomes. We have homologous chromosomes because we got one from mom, and we got one from dad. And those 2 sets of chromosomes came together to give us 2 of each chromosomes for every chromosome that we have. So again, 2 chromosome 1s, 2 chromosomes 2s, and so on and so forth. And then when we go on to have children, and we do, then the chromosomes that we have will get divided. We'll talk about this in meiosis. So that we pass on to the next generation half of our chromosomes, which is actually a mixture of chromosomes that we got from mom and dad. You don't give all mom, you don't give all dad, usually. It's possible, but it's highly unlikely. Homologous chromosomes, it basically says we have 23 pairs of chromosomes. So if you were to look on chromosome 1 and you saw there a gene for eye color, then you look at the other homolog, the other chromosome 1 you have that you got from your other parent, and you will find there a gene for eye color. That doesn't mean they'll have the same color. One might say blue eyes, the other one may say brown eyes. But you will have a gene for eye color on both of them. You may have a chromosome on one chromosome, there's a gene for the blood type of A. And then on the other chromosome, you have the gene for the blood type of O. Because A is dominant over O, we'll get to this later on, your blood type will be A. Yeah, A is dominant over O. So that's what homologous chromosomes are. They're not identical to each other, but they have the same genes for the same features on each other. They just may have slightly different instructions about what that feature should look like. So in mitosis, the cells start off with these homologous chromosomes. It starts off with a diploid status, and it ends in the same status. It's still this 2n status. And this particular cell has 4 chromosomes. That's the 2n status. So at the end, it has 4 chromosomes. All right, so prophase. This is the first phase of mitosis. And I saw right away, I said, they've got this label there. It says aster. What is an aster? You know what an asterisk is? You're reading a book or something and they put in one of these little things on it. A little star. And then at the bottom of the book, you'll see a little star, and I'll tell you what that star is talking about. So it's just a note place to tell you, hey, there's something that we can't fit into the book, so we put it at the bottom so you can read about it. Sometimes you use numerals. An asterisk basically means star-like. So they look at that, and they look at this, and this looks like an asterisk. It looks like a star. But it's not. It's really basically where the centrioles are. Those are the centrioles. The cell has 2 of them. And during mitosis, during prophase, these develop. And you'll see when you're looking at cells that are dividing, these asters on either side. And they look star-like because sprouting from them are spindle fibers. They're part of the cytoskeleton. They're microtubules made of tubulin. The other thing that's happening during prophase, other than these asters are separating, these centrioles are moving to opposite poles, is that the genetic material is condensing. The chromosomes are condensing, so we can see discrete chromosome bodies. Normally, during interphase, the DNA is all spread out in the nucleus. It's very active. It's making new proteins. But when we're getting ready to move. Think of it that way. We're getting ready to move. We're not moving to a new house per se. We're building a new house. The cell is getting ready to build two houses, and it's getting ready to move its DNA to both of those houses. And before you move, what are you going to do? You're typically going to put everything in a box, so you can carry it conveniently. It doesn't get trapped or caught on anything while you're moving it. Like a big curtain, you put it in a box. And that's what the DNA is doing. It's being put into a box. It's being condensed down into shapes that can easily move within the cell. So the chromosomes condense. And the nuclear envelope fragments. Here we have a nuclear envelope, and then here we have little pieces of it, because when you're looking at it on the microscope, it looks like it's just disappeared. But it's basically just being taken apart. The nucleolus, as we may or may not have recalled from the last unit, is where we construct new ribosomes. We put them together, ribosomal subunits. We're not doing that when we're getting ready to move. We're getting ready to move, so we're focused on the moving. So we don't want to put together proteins so the nucleolus disappears, so the ribosomes aren't being assembled anymore. The centrioles move apart. We're seeing here, they're close together here, now they're moving apart. And as they move apart, spindle fibers are forming from them. They're getting more and more spindles. I want to actually put my centrioles in my picture here. How are they doing their centriole? Sort of like this. There's my spindle fiber and the other spindle fibers, other chromosomes. And on the other side, we have another pair. We have spindle fibers here, spindle fibers there, here. So the spindles are forming. So metaphase, the book has it mentioned by both a pro-metaphase and a metaphase. Since each chromatid is attached to a spindle fiber. So I've already attached a spindle fiber to my chromatids here. Show you that point earlier. So every chromosome gets a spindle fiber. Every chromatid gets a spindle fiber attached, because we need to separate out the chromatids. And the opposite forces of the centrioles acting upon the chromosomes ultimately are going to push them into alignment. You know, pay careful attention to the pictures, because this is what you're going to be asked to duplicate during lab. So attachment spindle fibers, opposite chromatids at the centromere. And then the spindle fibers are all going to line up on the equator, and that's called metaphase. And this is what it actually looks like if you catch it on a microscope with blue stain. Inherited metaphase in its entirety, and we'll be looking for this on slides today in lab. So also worth noting, we've got this information for you. Once those chromosomes are lined up, the next step is anaphase. This is where we see the chromatids have separated. So if I were drawing this more to scale, My chromosomes always end up looking weak too. When we get to anaphase, basically what happens is that these get pulled apart. So this goes from here, and now the chromosomes are being pulled apart and looks like this. So these are metaphase, this is anaphase, but the chromatids have been pulled apart. Remember though, that these ultimately were, at the beginning, these were nothing more than duplicated chromosomes. So this is a chromosome now. And so is this. Free from its sister chromatid, it just gets called chromosome. And that's what this says. Separated sister chromatids become chromosomes. Once they're no longer attached, we don't use the word chromatid anymore. Telophase, the nuclear envelope and nucleolus reappear. The reason is because you've got all the genetic material pulled apart, and now it's in opposite poles of the cells are getting closer, and so we can bring back the nucleus to wrap around the DNA, and the DNA can start to relax again and not be condensed anymore because it's in a safe environment. It's been moved, so we're ready to unpack it. Then moving, time to unpack it. So the chromosomes relax. When the chromosomes relax and spread out, you don't see them as discrete bodies anymore. It just spreads out as a sort of this sphere inside the nucleus. And one of the key features that you look for when you're looking for telophase, to distinguish telophase from anaphase, you may not see the nucleus reappear. Well, the first thing you will see is this little dimple on either side. Not for anaphase. This is where the book gets it wrong. We use this as a reference point for telophase. So when you see that cleavage furrow, that's when you know you're looking at telophase. I make mention of this for when we're looking at slides today and you're trying to find telophase. You need to find that cleavage furrow to indicate that you're looking at this is a great telophase. If you can find one that looks like that on the slide today, aces. And cytokinesis, remember, is cytoplasmic division, is the splitting at the cleavage furrow. So all we do, as we complete that division, we completely go through until these are two separate cells. So cytokinesis is the final step. Cells become fully divided. Questions about mitosis? Alright, so what about meiosis? Where does meiosis figure into this whole picture? Mitosis is about repair, maintenance, growth. Meiosis is about the production of gametes. So what are gametes? Another word for gametes is sex cells, which basically, depending on which gender you're talking about, means either egg or sperm. The fancy word for this is oocytes for the egg, and sperm is spermatocyte. We all know what CYTE means. Cell. Ovo, ovum, egg, ova. Fancy words. Those are the gametes. So in order to produce sperm and egg, you got to go through meiosis. And the whole idea behind this is to go through two consecutive divisions. We want to produce 4 cells from our initial cell. And during this process, it's really critical that we want to go from that diploid state to a haploid state. So haploid HA for half. half the normal number of chromosomes. So for us, that equals 23 chromosomes. This cell has normally the diploid state was 4, so the haploid state is 2, half the number of chromosomes. And the reason we want to do this is so that when egg and sperm come together, we got 23 in egg, 23 in sperm, they come together and we make 46 again. That's the human condition. 46 chromosomes. You don't divide them by half, then you don't end up with normal cells. You get too many chromosomes. The other reason we do this is for genetic diversity. It may be hard to see it is because they're not really showing it in this picture. Each one of these cells is genetically different from the other one. And none of them are two are the same. Because you're actually giving different amounts of your parent's DNA to your offspring. You're giving, so one child you may give 90% of mom's genome and 10% of dad's genome to your offspring. And the next time that an egg or sperm you produce, you may give a 50-50 from your previous generation from mom and dad. And the same thing's happening with your spouse and then your significant other. And then when the egg and sperm come together, it's whole new combinations of genes that weren't there before. And the idea behind this for us is potentially to make it so that we always have enough genetic diversity in our population that in case there are changes in the environment, that not all of us get wiped out. And mostly for us, that has to do with disease. So when there's a pandemic, some people will die, but some people will live because they're just innately immune because they're not genetically the same. We are all genetically identical, and we all have the same weaknesses. When a virus happens to have the key to that weakness, it would wipe all of us out. But because we're not all the same, because diversity is strength, there's always the likelihood that some of us will be immune to any given disease that comes along. Even HIV, which was considered a fatality rate of 100% when it first appeared, if you got it, you were dead, and that's not true. Those two are equal. But they are relatively rare, so HIV is pretty significant that we take other steps to mitigate the spread. So we have these homologous chromosomes, and I've talked about these before already, so I won't repeat myself again. They are chromosomes where on one chromosome you have gene for eye color, and the other chromosome will also have gene for eye color. They may agree, they may both say brown, violet, they may not. And that's because we get our DNA material from our mother and our father. And half of them came from your mother, and half of them came from your father. We want to make sure of the two. Meiosis. So remember, 2 rounds of cell division, so we're going to have a meiosis 1 and a meiosis 2. And we're going to use the same phases, and a lot of the same sort of stuff happens through these phases as happened with mitosis. What we're going to focus now on is not what's the same, but we're going to focus on what's different. We'll talk a little bit about what's the same too. So during prophase 1, we would expect the nuclear envelope to disappear. And it does. Eventually, during prophase 1, this nuclear envelope will go away. But one of the things that's really important that happens during prophase 1 is that the homologous chromosomes come together. And when the homologous chromosomes come together, you do something called crossing over. And since you're going to be expected to replicate crossing over, it's kind of important that you think about this. So let's get rid of these chromosomes. Now we'll bring together a pair of chromosomes, say chromosome 1. So what chromosome 1 is going to do, I'm going to go ahead and move this up here like so. And then it's, remember, homologous pair, so this is from, we'll say dad, and then we'll bring over here another chromosome 1 from mom. And this is what happens. This chromatid will go and cross over here. And literally what happens is that we actually break these chromosomes in such a way so that we can now reconnect them in a whole new and exciting way. So they literally exchange parts of themselves. They genetically switch. This adds a whole other level of diversity. So again, my original explanation, like you get 80% from your grandmother and 20% from your grandfather, that's spread down, passed on to you through your parent, is true, but how come that's not necessarily as a whole chromosome? Chromosomes are literally bisected and cut and then reattached through this crossing over. Now note, when they are all together, when both of these homologous chromosomes are together, we have a total, if we count the chromatids, of 1, 2, 3, 4. And 4, another term for that is tetrads, or a tetrad. So when they come together, they call them tetrads, two chromosomes together like that. So these tetrads form all 23 pairs of chromosomes, and this crossing over event happens. During metaphase 1, just like in metaphase, chromosomes line up, but they line up differently. Instead of lining up in a way where all the chromosomes are end to end, we have now homologous pairs of chromosomes lining up. And so, and the equators right in between them, and the spindle fiber is attached to the duplicated chromosomes. They do not attach to each chromatid during this stage. You can see that the crossing over is occurred on the picture here. There's a little bit of red at the end of that blue, a little bit of blue at the end of that red. So the homologous chromosomes will line on the equator. And something to note is during this alignment, we get what's called independent assortment. Now you'll see that there's a blue and a red on this side and a blue and a red on this side. There's short blue, long red, short red, long blue. It could, however, have been short blue, short red, long red on this side. That's all independent assortment. Now, when they finish dividing, that means that this new cell is going to get one short blue, one long red, it's going to get short red, long blue. But if we get it the other way, then this ends up with two blues. And think about that. If you think about blue as being the paternal inheritance, then this cell would get almost all paternal inheritance, and would get almost all maternal inheritance. But this way, if we leave it like it is, and this cell is going to end up with about half and half. And this cell will end up with about half and half. So this independent assortment has an influence ultimately in terms of how much genetic information that you are passing down to the next generation came from your mother and your father. It's totally random when this happens. It can go either way. It's like flipping a penny and getting heads or tails. That's what independent assortment is. And this is just with two pairs of chromosomes. We have 23 pairs. 23 times you're flipping that coin for heads or tails. So the chances that you're going to send all of your dad's DNA to your child are almost zero. It means flipping the coin and getting heads 23 times. Unlikely to happen. So this independent assortment influences the genetic diversity of the sperm or egg that you are producing, and it gets passed on to the next generation. And then anaphase 1, how does it differ from mitosis? During anaphase 1, we don't separate chromatids, we separate homologous chromosomes. So the spindle fibers come into play here, and I'll use these as basically, we'll just pretend that I've got two different types of chromosomes here instead of being the same chromosome. And we put in those spindle fibers again, separated, we don't split up the chromatids now. This time, what we end up with, and I'm going to make it all blue at this point, the whole chromosome goes. And over here, we end up with the entire chromosome heading off. So very different from anaphase from mitosis. Homologous chromosomes separate. And the result of that, because they are duplicate chromosomes, is that the condition for these first cells during this first division is the haploid condition. They've already got half the amount of chromosomal material in terms of actual homologous chromosomes. They're all duplicated, but they're still half the amount of material. Telophase 1, the happens, we end up with the nuclear envelopes reappearing around the now half the number of chromosomes. We get cytokinesis, and we now have two separate cells. Sometimes this doesn't happen right away. Sometimes cytokinesis will hold off with the spermatogenesis, but we need to worry about the exceptions here. Chromosomes reach the poles, we finish off cell division. That phase between meiosis 1 and meiosis 2 is called interkinesis. I'm not sure why. It's in between the division. Because so interkinesis between meiosis 1 and meiosis 2 is between the two different types of meiosis. So what I want to say about meiosis 2 is meiosis 2 is mitosis, except for with just half the number of chromosomes. During prophase 2, however, we have to make new centrioles because we've already separated out the centrioles into two separate cells, and we have to make a whole new set in order to go forward with the second round of division. So the centrioles get duplicated. Spindle fibers attached to the chromatids. Just like in mitosis. Now, if we look at metaphase 2, the chromosomes align on the equator, but look, they're aligning in a way so the spindle fibers are attached to each chromatid. There are no more homologous pairs anymore. You've already separated those out. And now what we're going to do now is we're going to pull those chromatids apart just like in mitosis. So meiosis 2 is a replication of mitosis, but it's with half the number of chromosomes. So we don't reduce the number of chromosomes anymore. We're just separating out the duplicated chromosomes. We're separating the chromatids so that each of the final cells get a single set of genetic information, but we're at 23 chromosomes instead of 46. We're already at the haploid condition. So the chromatids separate, and when that happens, they become chromosomes. So as soon as it happens, they become chromosomes, and each of the new cells will get 23 chromosomes. So what about telophase 2? Is it exactly like mitosis? Absolutely. Spindle apparatus disappears, the nuclear envelopes reappear, the genetic material can now relax, and now we have potentially 4 different gametes. Cytokinesis has happened, maybe. We won't have to worry about that. But ultimately it does. So again, meiosis is about creating genetic variation. And we can see here a close-up of what I drew poorly on the board. They have 2 sets of chromosomes coming together, and here they've conveniently labeled them for you. So there's a capital D, capital Z, capital B, capital A, and lowercase a, lowercase b, lowercase c, lowercase d. They've done this because when we do genetics, it's a very common way of distinguishing between genes, using an uppercase and a lowercase. Uppercase usually means dominant, lowercase usually means recessive. So in this really oversimplified example, they're saying there's 4 genes on a chromosome. In reality, there's hundreds on a chromosome. And when we do this crossing over event, we end up with, instead of having all capitals on one side, we have a mixture now. We have capital B, lowercase c, lowercase b, capital A. So the crossing over has occurred, and the blue is now part of this red chromosome, and this red is now part of this blue chromosome, and it now has the capital B and capital C. So there's a development of genetic diversity within the chromatids, and then ultimately, the cells that are produced in meiosis, each one gets a different one of these. So one cell would get all the capital letters of the red variety, and one would get a little bit of red and then a little bit of blue. And then this 3 would get the capital C and capital B, and the lowercase a and d, and then 4 gets all lowercase letters. So none of these are identical to each other. They're all genetically different. Where you can say, this has got capital D, that's got capital D. Yeah, but this chromosome has little lowercase c. And this one has capital C. This has lowercase d, but there's capital C and there's little c on this side. So they're all different. And that's just with one pair of homologous chromosomes again. We've got whole 23 pairs. So between independent assortment and genetic variation, the amount of genetic diversity you get in gametes is astronomical. So remember, crossing over occurs during prophase 1, during tetrad formation. Independent assortment is also part of this. This image is supposed to show that to you instead of using just 2 pairs, they've got 3 pairs of homologous chromosomes. Here they've got large, medium, and small. I don't know why they didn't do it in order. But in this example, they have the large and the medium red on the left-hand side and the small blue on the left-hand side, medium blue, small red, and large blue on the right-hand side. Over here, we've got medium red, small red, large blue, where we have medium blue, small blue, large red. And then here we have medium blue, small red, large red. So every one of those is a different setup. Here we have all the reds on one side, all the blues on the other side. So that's 4 different setups with independent assortments with just 3 pairs. And again, there's 23 pairs of chromosomes in human cells. So the arrangement, just through independent assortment of creating genetic diversity is huge without even going into crossing over. Both are involved, both are exploding. That's why kids don't look exactly like their parents. Or anything else? The strength of genetic variation and change in environment. As I mentioned, this genetic variation protects us from future disease. It may protect us from future other changes in the environment. There are organisms in the environment that basically live their life in the haploid state. Remember, haploid means half the number of chromosomes. There are fungi, mushrooms out there that their everyday life is always haploid. The only time they go diploid, the only time they make homologous pairs of chromosomes, when they get together with another fungus when there's a change in the environment. And then they will go in through they will get into the diploid status by getting together with another fungus, and then they will do independent assortment, and they will go through meiosis in order to create whole new sets of genetic variation for the next generation, so that in this change environment, say there's a drought, they're more likely to produce a fungus that is able to survive the new environment. They only do this when the environment changes. It's an obvious adaptation to a changing environment to create genetic diversity, because why do it unless there was a threat to, if things are working as it is, why change it? If it's not broken, why fix it? So fungi then in a stable environment, they don't do anything but just mitosis. They make new copies of themselves by producing spores by mitosis. The spore goes and produces new fungus, and they keep on living happily. But when the environment changes, like, there might be new stresses, we may need new combinations to survive this new environment. They go through this variation, they create new spores with new features and characteristics so that they're more likely to survive. Maybe not all of them do, maybe one has the mutation, or I should say the variation that will allow them to survive in the new environment. And of course, we do meiosis in order to keep the chromosome number the same. We don't, otherwise, if we don't go through meiosis, our egg and sperm would have 46 chromosomes, and the next generation would have 92, and the next generation of that would have 184 chromosomes, and then we have 368 chromosomes. That's not going to work. We get a chance to talk about genetic abnormalities when people get too many chromosomes. Basically, usually results in a non-viable child. So we have to keep the number of chromosomes the same. It just doesn't work otherwise. So I ended this last time around here, so I'm going to go ahead and end it here today. That's meiosis. What we talked about. So I'm going to be back here for lab at 1 o'clock.